Hi everyone, today we'll be talking about the naked mole rat, also known as the Heterocephalus glaber. Some background information about this rat is that the naked mole rats are hairless wrinkle rodents. They have historically been found in Uganda and Tanzania. Their natural habitat includes underground grasslands, savannas, and other tropical regions. These rodents are generally brown and pink. They generally live about 28 years and feed on plants like bulbs and tubers. These animals have been around for a long time and are considered the only true aerosocial mammals to live in underground burrows in East Africa. They are the longest living rodents in the world. These animals are not completely hairless. They have whiskers on their face and tails for sensory purposes. They also have hairs between their toes that allow them to sweep and dig up soil. Their class is Mammalia, their order is Rodentia, their family is Bathydia, and their genus and species is Heterocephalus glaber. Some fun facts about these rodents are that they live deep underground in dark tunnels, they have poor eyesight, they have very loose skin which helps with them passing in and out of tunnels and amongst each other. They are mammals, they are the longest living rodents known to man, and they are immune to cancer and can live with very little oxygen. Their body temperatures also change with their environment. Naked mole rats cancer resistance is the most interesting part about their being. Cancerous cells introduced to the naked mole rat did not grow cancerous. This is very different from what is generally seen in mice. Findings show that naked mole rat cells are not more resistant to cancer than cells of other humans, but it's complicated in understanding why this is so. Mammals have evolved anti-cancer defenses consisting of cell cycle checkpoints called apoptosis and replicosensins, controlled by a network of tumor suppressor genes such as P53 and RB. Hypersensitivity to contact inhibition is called early contact inhibition, which is the key anti-cancer mechanism that prohibits cell division at high densities. Human cells use replicosensins as an anti-cancer mechanism, while mouse somatic cells express telomerase. In humans, both RB and P53 tumor suppressors play important roles in controlling cell proliferation, creating backup barriers for cancer progression, while in the mouse, RB plays a relatively minor role. As a mechanism, it has been found that critical genes in P13K pathway, including AKTL and PIK3CA, are downregulated in naked mole rat cells. Cancers are caused by mutations attributable to both intrinsic and extrinsic factors such as uranus, DNA replication, and environmental mutagens. Mammalian species in larger body mass and longer lifespan have an increased risk of cancer because such species have a greater number of cells and more time to accumulate cancer-causing mutations. However, in reality, larger and long-lived species have a lower incidence of cancer than small, short-lived species, which are known as Peto's paradox. So why the difference? Genes such as polymerase protein complex, which helps maintain chromosomes, were different in naked mole rats, which can possibly explain why their cancer immunity is so high. In conclusion, there are several genes that play an active role into why cancer rates among naked mole rats may be lower than that of other animals. The alteration of these genes has a huge impact on how they express and the vital balance between tumor suppression and proliferation in these animals are vital to the longevity of their lives. Mice with overzealous tumor suppression, which leads to an overactive P53 gene, which may lead to premature aging and a short-lived lifespan that can increase their risk of cancer. So this is really what differentiates the naked mole rat from other mice and other rodents in general. I hope you guys enjoyed this presentation. Here are my references and have a good night.